So this presentation is the first of a three-part series which I've called NGS Library Construction Theory and Practice. And so in part one, which will take more or less an hour, we're going to step back a little bit, look at some of the high-level theory behind library construction to sort of provide context for what comes next. In part two, we'll start digging into more of the practice of it and looking at different library construction parameters and how to optimize each of these to make the best possible library from your available sample. And then in part three, we're going to get really, really practical and really go step by step through an example of how to make a library with the Kapa Hyper Plus library preparation kit. This is a very exciting new kit, which is officially actually being launched today. And so at the end, hopefully at the end of the seminar, we'll get some of this, some of the more, we'll get some of the more get into some of the practicalities around library preparation, and hopefully have you leave with some really good guidelines of getting right into it. And so switching straight into part one, what we'll cover in here is a very, very high level overview of Illumina sequencing and applications and technology. This is not trying to be an explanation of Illumina sequencing technology. There's a lot of good information on the internet about how it really works. And I believe that you guys will also have a Illumina dedicated seminar later in the series. But it's just to provide enough context for what follows. And so during the course of this first part, we're going to keep on answering this question: why do we do library construction? And so from a practical point of view and also from a more philosophical point of view, we'll briefly look at different library construction strategies, and then end with the question to lead into part two is what is the best library I can make? And then in part two, we'll continue say, but how do I make that best possible library? And so on a very, very high level, Illumina sequencing is a next generation sequencing technology that allows us to sequence DNA. And so we can either sequence new things, which is called de novo sequencing, or we can sequence things that's already been sequenced, which is broadly referred to as resequencing. And so in terms of things that we can sequence, obviously the largest genetic units we can sequence are whole genomes. And so we sequence a lot of humans lately. That obviously has a lot of impact on human health and reproductive health. We sequence model organisms. That helps us to understand human disease and provides better and new and targeted treatments for human disease. We sequence emerging pathogens, so things that affect the quality of our life, and crops, the things that provide us food. And so because whole genomes are big and they're expensive to sequence and they generate a lot of data, a lot of especially resequencing is called targeted sequencing, so where we sequence smaller units of the genome. So for instance, we can sequence just the exome, so all the expressed genes really in the genome, or we can sequence anything that's called a custom panel. And so for instance, we can look at all the genes associated with certain diseases. In the clinical world, they talk about actionable panels nowadays. So these are regions of the genomes that are associated with known treatments or treatments that are in trials at the moment. So lots of different ways of looking at smaller subsets of the genome that are cheaper, easier to turn around, and provides more targeted and actionable information. Then there's a category which I just call specialist sequencing. These are technologies or techniques that help us look at very specific things. For instance, ChIP-seq is a sequencing methodology that helps us look at DNA-protein interactions. We can look at epigenomics or study epigenomics. A technique called methyl-seq that allows us to look at DNA that's specifically methylated in genomes. And then I've sort of also grouped metagenomics under here. Many of you may have heard of the human microbiome. It's a very sort of hot new topic to look at all the microbes that live in our gut and how they affect human health and the quality of our life. And so there's lots of sort of different specialist sequencing techniques. And although Illumina sequencing is a DNA sequencing technique, we can turn RNA into DNA, into cDNA, and that also allows us to do RNA sequencing. Not directly, we don't directly sequence RNA molecules, but through the cDNA intermediate. And so that allows us to look at gene expression, so the different things expressed at different times in different cells in the body or in different populations of samples. It allows us to look at gene structure, for instance. you may have heard of splice junctions and the structural variants of genes and isoforms that have important implications, again, for disease and health. And we can look at gene regulations through things like small RNAs, microRNAs, and all of those sort of non-coding RNAs other than tRNAs in the body. So those are sort of, on a high level, the sequencing applications. And then I just line them up with it's sort of the sample types that we're going to use for these different applications. So again, starting with whole genomes, we can sequence, genomic DNA, cell-free DNA or free circulating DNA or circulating tumor DNA. This is a sort of very interesting group of DNA species that's naturally fragmented by the body and kind of lives in our blood. And are good indicators for early indicators of disease, our state of health, and so becoming a very interesting sample type sequence. FFPE DNA, formalin fixed paraffin embedded samples are used very much for clinical applications. So many patients will come in, they'll have a biopsy done, the tissue will be fixed and used for histology and pathology, but we're also trying to get the genetic information out of those tissues. There are lots of other types of clinical samples and even single cells. And so we can start sort of with samples that represent the whole genome. From there, we can generate the material for targeted sequencing, either through what we call target capture. So in that case, we'll make a whole genome library and then use a set of hybridization probes to just catch or select the DNA that we're interested in sequencing. Or we can use PCR and specific sets of primers to target the subset of DNA that we want to sequence. Then for the specialist applications, for ChIP-seq, we generate DNA through chromatin immunoprecipitation. That's a technique that where we use antibodies to pull down basically the DNA sequences that interact with specific transcription factors. We can make, again, a whole genome library, treat that with bisulfite, and that converts only those cytosines that are methylated to uracils. The rest are non-methylated to uracils. The methylated ones stay at cytosines. And so we do methyl sequencing and study epigenetics through bisulfite converted DNA. And then there's a host of environmental samples, all kinds of soil samples, clinical samples that we can use for these specialist applications. And then for DNA, for RNA, there's essentially three types. So total RNA, mRNA, or small RNA. And the library construction methodologies for those differ a little bit. mRNA and total RNA, pretty much the same and a very different way of making small RNAs. And so right up front, I want to point out that most of what will come in the rest of the seminar series is very much focused on DNA sequencing. We have a very, very nice webinar on RNA sequencing and library construction for RNA sequencing. So please visit our website and go listen to that if RNA sequencing is your specific application. Irrespective of the sequencing application and what it is that we're trying to sequence and for what purpose, the objective of Illumina sequencing in the end is to generate millions and millions of short overlapping sequences. And from those short overlapping unique fragments, we then assemble the DNA sequence, the entire DNA sequence, whether that's for the whole genome or for a part of it or for the expressed genes or ChIP DNA. And so that's really the objective, and that's what I sort of want to keep in mind when we talk about library construction, that we always keep in mind that that's the objective of sequencing in the end. There's lots of sequencing metrics and QC metrics and terminologies that's important, but I just want to sort of highlight three for the purpose of this series. The first is sequencing read length, and that's basically, it's essentially the length of these determines the length of these overlapping fragments. And sequencing read length is determined by the sequencing chemistry you use, also the instrument you use. With the Illumina sequencing, it started off very small, 20 or 30 base pairs at a time, and the technology has improved quite intensively over the last few years, allowing us to go, do sequencing read lengths or single fragments up to around 300 base pairs on a MySeq, and 125 to 150, 250 on a HiSeq, depending on in which mode you run it and which chemistries you run. The 2x just means that we do paired end sequencing, so we sequence from both sides of the molecule, and so for every every library molecule every unique fragment that we generate, we're able to read from both ends. And sequencing read length has an impact on many things, many aspects of library construction. That's why I wanted to point that out. The really important sort of concept, and again that that answers or helps us answer these questions about why do we do library construction and how should we do library construction, has to do with coverage. So coverage, in a very simple definition for coverage, is essentially the number of unique overlapping fragments that's available for every single position that you're trying to look at. And so it has to be unique fragments. The sequencing technology works such that if a fragment has the same start and stop site, it's seen as a duplicate, an experimental duplicate, not something that exists biologically, and so it doesn't count. You have to have unique fragments. And so the coverage depth depends on a little bit on the application. So for instance, for whole genome sequencing, you may aim to have to cover every position at around 30 times. Obviously, the deeper the coverage, the more confidence we have in the amino acid identity at that position. And for some of the clinical applications, coverage depth can go up to 100, 250, some 500, 1,000-fold. If, for instance, we want to with confidence identify small, very rare cancer populations against a background of normal cells, or we're looking at very rare allele frequencies, rare mutations and rare allele frequencies associated with different diseases. The strategy there is to sequence a lot, smaller genetic unit, but sequence that to much, much deeper coverage. And so coverage depth is really important, and different types of projects have different requirements. But what's even more important is coverage uniformity in the end. And so, the objective of our sequencing, so the first way in which we can answer this question is, what are the objectives of library construction? The objective really is to get every position that you're interested in covered, as deeply as you can or as needed, but to the same extent. And so, whenever you have a sample and you end up, that you need to prepare for sequencing, this needs to be the objective. When you have situations as the ones portrayed below, where there's regions of the genome that are covered very sparsely, so the trick here is not to see how deep you can cover any one base, but how uniformly you can cover every base. And if you have regions, and the reasons we see low coverage in certain regions is because of the structure of DNA, inefficiency or limitations of library construction technologies, cluster amplification, the sequencing technology itself, and even some of the analysis aspects. And so whenever we are missing regions or have regions that are covered below the threshold that what's needed for the objectives of the project, it means we have to sequence more. And sequencing more obviously costs more. It also increases turnaround times. And in worst case scenarios, we may have to use completely different techniques like Sanger sequencing to fill those gaps. And so really, as I said, the first answer to this question of, what is the objective of library construction? It is to make, a library that allows us to get the highest and most uniform sequence coverage for every single position in the sequence region that we're interested in. And so having looked at sort of what the high-level objective is, I'm going to move into just looking at Illumina sequencing technology on a very high level. And essentially, the process from sample to sequence, happens in 5 stages or 5 distinct steps. The first one of these is fragmenting the DNA. So obviously, Illumina is a short-read sequencing technology. We're sequencing short DNA fragments. And so whatever sample we need to, we start with, if it's not already fragmented, like cell-free DNA, then we have to fragment the DNA. And there's different ways of achieving that, which we'll discuss in a lot more detail. But essentially, there are enzymatic ways of fragmenting, and there are mechanical ways of fragmenting. And as I say, we'll go into that in more detail later on. We then have to define the molecules that we want to sequence, because the whole idea of fragmentation is to break the DNA that we're interested in into random pieces, completely random pieces. Remember, we're trying to get unique overlapping sequences, and so fragmentation has to be entirely random. We don't know how to deal with these molecules. They all have different kinds of ends and middle parts. And so what we do to define these molecules and basically get a handle on them for the next steps of the process is we add what we call adapters to them. And so that's the process that we call defining the molecules. These adapters or handles then allow us to do to isolate and multiply these molecules. So all next generation sequencing technologies are aimed at sequencing individual DNA fragments. But the sensitivity of the technology is not high enough to get enough sequence out of individual molecules. So for every unique fragment, we have to multiply that fragment. We have to make sort of a population of a clonal population of each fragment in order to get a strong enough sequencing signal. In Illumina sequencing, this is done on a two-dimensional surface called a flow cell, and so the surface is populated with a bunch of small primers, which are complementary to the ends of these adapters. And through this process called cluster amplification, we generate for each individual unique fragment basically a little colony of fragments or a little clonal population of fragments, which just helps to intensify the signal from that unique molecule during sequencing. Next about is sequencing. So Illumina uses sequencing by synthesis technology and very sophisticated imaging technology to essentially generate a sequence of light signals that are transposed into actual sequence. And then the final part of this is the sequencing analysis. And so this is the process where we take all the raw sequencing reads, trim the ends, align them, and then go through all kinds of different bioinformatic processes to derive the information we need from the sequencing. And so really, why do we have to do library construction? It's pretty obvious from this process. Because we have to sequence short fragments, and we have to do cluster amplification and sequencing, we need some way of anchoring known sequences to unknown random sequences. We have to do library construction, and library construction really encompasses these two top stages of the process, which is fragmenting the DNA and then defining the molecules by adding adapters. And so let's look a little bit at the two, those two different aspects, fragmentation and then adapter ligation. Why is fragment length important? It's important for a bunch of reasons. So first of all, the technology, the way the Illumina technology works, there are limitations on the sizes of molecules that can be accommodated. So sequencing read length, the sequencing chemistry itself imposes some kind of a size length, but that's improved greatly over years and is less important. The important thing is that the cluster amplification process doesn't work very well for molecules more than about 700 to 800 base pairs. And so anything that we don't get into that size range basically can't be clustered and can't be sequenced, and we lose that from the population of molecules. So that's the first reason why fragment length is important. Then it's also important for a whole host of other reasons. So we've talked about sequencing read length a lot. For instance, if you're going to be doing 2 by 150 sequencing on a high-seq, it depends a little bit on your application. If you are doing, for instance, de novo microbial sequencing, you probably want to have your inserts, the fragments that are defined by adapters, a little longer than 300 base pairs, so that you can make the most out of your sequencing capacity. So you'll have a read of 150 base pairs from the one end and 150 base pairs from the other end, and so you sequence almost the entire molecule from both ends without having too much of an overlap in the middle, so that you're essentially losing sequencing capacity there. On the other hand, if you're doing clinical applications, for instance, sometimes there are cases where the first read and the second read are used to confirm the sequence, and then it's better to have a sequencing fragment length, a library fragment length around 150 base pairs, so that you can read through the entire sequence from both ends and get a 100% overlap. Another example why it's important for application, for instance, is exome sequencing. The exomes, the expressed coding regions of the genome are on average 150 to 200 odd base pairs long. And so, if you're preparing a library for exome sequencing, normally you prepare a whole genome library, then pull down the exomes with hybridization probes. If you've got a really long library molecules, what are you going to be pulling down? Statistically, are a lot of non-coding DNA that just ends up in your library, and that's stuff that you want to throw away. And so you tailor your library size to the to the sequencing application and the nature of what you want to sequence. In some cases, it's very important to have a very uniform fragment length in your library. So, for instance, for de novo sequencing, where there's no reference genome to align your sequence to, if you have to build that genome from scratch, then the whole bioinformatic process is a lot easier if the fragment lengths are more consistent in size, it's easier to then do an assembly from that. So and also larger fragments, they are better than just a whole bunch of really small ones. And then finally, sample type. This is important in the clinical space. For instance, FFPE DNA, so DNA that comes from formalin-fixed blocks. The fixation of the DNA of the tissue, as well as the extraction of the DNA, is pretty destructive. And so we end up with DNA that has a lot of molecular damage of all kinds of sorts. So we have oxidation and deamination and cross-linking and stuff like that. And those samples can typically not sustain library amplification more than 200 or 300 base pairs. And so trying to make really long insert libraries out of FFPE DNA is a stupid thing to do, because you're just going to lose too many of your fragments during library amplification and during, during cluster amplification. And so for FFPE, typically, people are making insert libraries in the, 150 to 250, 300 base pair range, and doing, 1 by 2 by 76 or 2 by 100 sequencing read lengths, rather than 2 by 250, which makes no sense for that sample time. And so in summary, what's important is, to optimally use your sequencing capacity, it's important that you pick the most appropriate fragment length for, the sequencing read length, or your sequencing chemistry, your sequencer, your sequencing application, and your sample type. And then when you do fragmentation, when you pick a fragmentation method, that you pick one that gives you, the ability to tune your fragment length, obviously, but also good control over your fragment length in the end. Okay, so now we've looked at fragmentation. The other part of the library constellation process is that process of definition or adding adapters. Why are they important? So what we've depicted here is essentially, just a random DNA fragment that becomes part of a library, flanked by 2 Illumina adapters. The Illumina adapter has a bunch of functional elements, and what I've depicted here is sort of the latest generation of dual indexed adapters. So if we go sort of from the outside in, just looking at these different functional elements, right on the ends of the molecules, on the 5' end and the 3' end, and the reverse for the complement, they are what we call the P5 and P7 sequences. These are sequences that are needed for cluster amplification, and so the flow cell, as we said before, it sort of has this lawn of oligos that are complementary to the P5 and the P7 sequences. And these are the sequences that we need to anchor molecules to be sequenced to the flow cell for cluster amplification. And so by the time that you want to sequence your library, that you're ready for sequencing, the adapter has to be completed. You don't have to add all these sequences in one shot. In most Illumina library construction protocols we do, but you can also build this adapter bit by bit. Important thing though is, by the time you're ready to sequence, all these functional elements have to be there. Sort of internal to the P5 and P7, we have the areas to which the sequencing read primers bind. And as I mentioned before, you can, with Illumina sequencing now, it's very common to do paired end reads, so you sequence the molecule from both ends. And so the read 1 sequencing primer is complementary to this area, and the read 2 sequencing primer to this area. And essentially, once you've made your clusters, you're going to first do the first sequencing read. You essentially then denature everything, regenerate the cluster, and then you do the read 2 sequencing. So sequencing primers. Now, there are applications that will just do single end reads, and then you can either, only just do the one read, or use an adapter that only provides for the one read. And then the third one is what we call the index, or the barcode, and this allows us to do multiplexing, so pooling of libraries before capsule sequencing. This again, so the degree to which you do multiplexing depends again on the sequencing application, the sample type. For instance, if you're sequencing whole human genomes, you're typically going to load, a single sample on a single, lane, and then there's no reason to, to distinguish samples, in the final sequence data from one to another. If, for instance, you're sequencing microbial genomes, you can sequence up to 1,000 on an Illumina flow cell, or on a single lane, depending on the size of those genomes. So some microbes are pretty small, and you can put a lot of them in there and still get your 30 or 50 or 60x coverage that you need. And so, if you're going to mix samples for sequencing, you have to have a way to identify which sequencing reads, belongs to which sample in the end. So demultiplexing everything at the end. And this is why we have these barcodes. And so traditionally, we were working with single barcoded Illumina adapters, which would just be sitting just so between the V2 sequencing primer and the P7 oligo. Nowadays, it's possible to and commonly used to use an index on both sides. That allows us to multiplex to a larger extent with less complex oligos in a way. And then you get other variations too. I mean, there's also a trend towards using what people call molecular barcodes. This is a different type of barcode that identifies molecules right from the beginning of the library construction process. And that's a bunch of unique random barcodes that allows us to essentially distinguish between molecules that are duplicated in the biological sample versus molecules that are duplicated as a result of library construction. I'm not going to go into too much detail. But essentially those three elements, so the ones on the outside we need for cluster amplification, the ones on the inside right next to the DNA insert we need for sequencing, and then we can any kind of barcode design depending on your sequencing application, sequencing coverage and capacity, that can be designed whatever you require. Important if you want to pool samples, so in target capture you can pool before you're even doing the capturing. It's important that you need to have barcodes at the time that you're starting to pool. So if you're not, if your library construction process isn't designed such that you're adding full-length adapters, you need to make sure that all the functional elements are in the right place at the right time during the process from sample preparation to sequencing. Okay, so now we kind of talked a little bit about fragmentation. We talked a little bit about adapters and the functional elements, and so I want to switch next to different ways of making libraries. So we know we have to create fragments, and we know we have to add adapters. And so the simplest technique to create libraries is by PCR. So you all know that we can use the PCR technique to amplify or isolate specific fragments of DNA. And so we can use specific primers to target only those regions of the genome that we want to sequence. Obviously, this is only for targeted sequencing. We would never do this for whole genome sequencing. It would be kind of stupid to try and generate a PCR pool of the whole genome. But there are definitely specific applications where generation of amplicons or making that pool of fragments by PCR is the appropriate thing to do. And then we can add, so in this picture, the yellow bits would basically be, the blue bits would be the gene-specific regions, the yellow bits would be sort of a little overhang that starts building the adapter, and then we can add the rest of the functional elements by another round of PCR. So typically, the first one would be a multiplex PCR, or we would pool the products from a whole lot of individual single-plex PCRs, and then just complete the functional elements in the adapter by another round of PCR. There's lots of different variations on this. We can use really long primers from the beginning that contain all the adapter sequences. It all depends a little bit on the application. The second way of generating libraries, so fragmenting DNA and adding adapters, is by fragmentation and PCR. So this employs things called transposons, which have been known in molecular biology for many years. A transposon is essentially a mobile DNA element that inserts itself in different places in DNA, and there are enzymes called transposases that carry these DNA elements and allow them to insert into the DNA. What's really nice about fragmentation technology is that it fragments the DNA and starts adding the adapter in one step. So it's kind of a convenient way of doing it. You don't have to worry about an individual fragmentation step. And so essentially the transposase inserts into the DNA, creates these sort of fragmented pieces of DNA that now carries the little stubs of the adapter, which is then completed by PCR. The third way of doing library construction is by a combination of fragmentation and ligation. And then PCR may or may not be needed. So the fragmentation can be done either through mechanical means. All of you have heard about a Kapa instrument. That is an instrument that uses sound essentially and mechanical shearing to fragment DNA very randomly into short pieces, and the fragment length can be controlled through the intensity settings and different settings on the Kapa instrument. Another way of doing that is by enzymatic means, and so we use enzymes to cut up DNA. Up to recently, that's been very hard to do, just because it's very hard to get enzyme mixtures that fragment DNA in a completely random fashion. And we're very excited about the product that we're just launching this week, which contains an enzymatic fragmentation reagent that now is capable of complete random fragmentation of DNA. Then for Illumina sequencing specifically, the fragmentation process is followed by 3 enzymatic processes. The first of which is called N-repaired, and so because fragmentation leaves the ends of DNA kind of frazzled a little bit, we fill in these ends and make sure that they're phosphorylated to create sort of blunt-ended DNA fragments. These fragments are then A-tailed. The only reason they're A-tailed is because the Illumina adapter uses TA cloning. Essentially, it's just a way to make the ligation process a little more efficient than blunt-end cloning. And so we first have to create blunt ends, 3 A-tail them, and then we ligate through a process of double-stranded DNA ligation. We ligate DNA fragments with an A-tail to adapters that contain a T-tail. And as I said, the TA cloning just makes the cloning a little more efficient than blunt-end cloning, blunt-end ligation. And you can see that the Illumina adapter has sort of this Y structure. So for ligation-based library construction, has a Y structure that can. It's a single adapter that can either ligate on the front or the back end of the DNA. So it's sort of a mirrored structure, and it creates the same in the end. The same ends on both sides of the molecule, the same functional elements on the 5' side and the 3' side of each molecule. And so then we use the process of library amplification. If the adapter didn't carry all the functional elements we need, so you can ligate a truncated or a shorter adapter. We complete the adapters by PCR, or we use PCR to make more molecules for the next step of the process if needed. And so, in fragmentation and ligation-based library construction, library amplification, depending on the adapter design, may be optional. And there's also sort of a combination of things. For instance, you can use PCR to generate multiplex, do a multiplex PCR to generate DNA fragments. But instead of completing adapter elements by PCR, you can then ligate adapter elements to them. And so, these are sort of just in broad strokes the 3 processes for creating DNA libraries. There's also specialist steps. For instance, in methyl sequencing, there's a bisulfite conversion step. For RNA sequencing, obviously, before we get into the DNA library construction part, we have to convert RNA to cDNA, and that's a completely different process. And so, knowing about all these different ways of making libraries, we get back to sort of this key question: what really are the objectives of library construction? So, on a physical level, on a practical level, it's about fragmenting DNA, and it's about ligating adapters. But with so many different strategies of doing that, and so many different kits and things out there, remember the original question is the objective of library construction in the end is to make a library with the highest and most uniform coverage there. And so, when we have our sample in our hand, we always, and we need to make a decision about how to make this library for the sequencing application and the biological question that we're trying to answer. The things that are sort of, close to us is how much DNA do I have? How much DNA do I need to make the library? To what size need I fragment it? We discussed that in detail. And then in the end, we make the library and say, did it come out the right size? Sure. Do I have enough? And so really, I mean, on a basic level, most people are just interested in, from the material I have and, with the kit and the method in hand, can I make a library? And what we're trying to, instill is to not just answer that question, but to really answer the question is, how do I make the best possible library? If I've made a library, how do I know that's the best possible library I could have made? I mean, obviously, we're not going to make the same library over and over and over and sequence it over and over and over to answer that question. And so, there are definitely some, theoretical considerations and some education up front to answer this question of, if I only have the sample and I have one shot at it, how do I ensure that I make the best possible library? In other words, the one that has the highest coverage depth and the most uniform coverage for every single position that I'm interested in. And so with this in mind, these are the three elements that we need to consider. And so the best library is obviously the one that has a lot of unique fragments covering every single position and a similarly high number of unique fragments for every position. So coverage depth and coverage uniformity. And to achieve this, what we need to look at, and as we consider different ways of making libraries, what is the most random but even way of fragmenting the DNA? Obviously, if we use a fragmentation method, and this includes PCR, because remember, we can create those fragments during PCR, are we doing this in the most random possible way? If we don't do it in a random way, we're going to end up with duplicate reads, and they're going to be thrown out and be of no use to us. What is the most efficient way to attach the adapters? That sort of helps us determine how much of the DNA that we have in the beginning are we able to convert to adapter ligated library. And we talk about the sort of the diversity or the complexity of the library, and that ultimately again determines how many unique fragments do we have covering every position. And then if we do have to do library amplification, so that might either have to be, is either mandatory for the method we choose or is optional, but may be determined by adapter design. If we have to amplify the library, or if we just kind of need more for the next step in the process, we have to make sure that we do it in the most efficient and unbiased way. And so if we look at, go back to these different library construction strategies just on a high level, looking at sort of the advantages and disadvantages of different ones of them. So PCR is by far the fastest and most convenient way of making a library. I mean, you can complete library construction, especially if you're using primers that contain the full-length adapter sequence. You can essentially make a library in less than an hour with very little mess and fuss about it. But why don't we just all do that? Because PCR is biased. PCR is biased in terms of fragment length, so shorter fragments are always amplified easier and to a larger extent than longer ones. And it's also biased in terms of sequence content. So all enzymes battle to amplify GC-rich and AT-rich regions with difficult secondary structure. And because of the exponential nature of PCR, this bias kind of gets amplified. So in the first round of PCR, if there's something that you're missing or underrepresenting, it just completely drops out during the number of cycles that you need to generate enough material for the next round. Also, another thing about PCR is that it's difficult to do a multiplex PCR with high fidelity enzymes. It's just the nature of high fidelity enzymes, and because we are sequencing individual fragments and not populations of fragments during next-gen sequencing, fidelity is important. So we don't want to introduce nucleotide substitutions during the process of generating the fragments that we need to sequence. Next, in terms of sort of speed and convenience, is fragmentation. Again, you don't need any specialist equipment. The fragmentation happens at the same time as the start of library construction. But fragmentation does have some serious setbacks. It is less random than other fragmentation methods. Fragment length can be really variable with fragmentation, so most fragmentation-based kits are very sensitive to input DNA. And if you get that input DNA a little wrong, then you end up with a very, very broad library distribution. And that either means that you're going to need to size select fragments out of there, so you lose a lot of material that you end up not being able to sequence. Or when you get to cluster amplification, some of that just doesn't sequence. And so control over fragment length is definitely important for successful and efficient sequencing, and that can be a bad setback or disadvantage of fragmentation-based methods. Something that's a little subtle about fragmentation is the way the adapters are added, so that the transposon, when it inserts in the DNA, it can basically go either way, and so you end up with half of your molecules with what we call like a AA or BB configuration. So it has the same adapter on both ends and not a unique adapter on either end. And so even if fragmentation was 100% efficient, you'll only get 50% of the molecules that are sequenceable, and that's something to consider when you compare it to other methods. And then library amplification is mandatory. So because the transpose size carries just a very small stub of the adapter has to be completed by PCR. And then depending on the PCR enzyme that's available for the method or the kit that you choose, that amplification can be biased and can contribute to uneven coverage and dropouts in the end. And so third, least convenient and the longest, traditionally is fragmentation and ligation, where fragmentation is an independent process that up to, recently, Kapa sharing that requires specialized equipment and specialized consumable and is pretty laborious, was the only good way of fragmenting DNA. Again, if you stay all the way until part 3 of this series, you'll see that we've got great alternative enzymatic solutions available to us today. But fragmentation, Kapa sharing, and then also the new enzymatic reagent that we've developed is less biased. It's the least biased way of fragmenting DNA. It's the way that gives you most control over fragment size in the end. So either through the fragmentation conditions or size selection in the end. Ligation can be very efficient, and so we'll talk about ligation a lot. It can be very efficient if your inputs aren't limiting. And one of the advantages of this sort of strategy is that library amplification is not mandatory, and it can be limited, depending on the details of the process. And so, traditionally, although fragmentation and ligation, is the least convenient, longest turnaround times, it is definitely the strategy, the library construction strategy, that leads to the best performance, or the best library quality and sequence, best use of sequence capacity in the end. And so what we've been doing at Kapa is focus very much on ligation-based library construction, and our efforts in recent years was to achieve a combination of really good performance, but with make library construction faster and more convenient. And again, the HyperPlus kit that we've just launched really combines all these aspects of high performance, less biased fragmentation, good control over fragment size, high library efficiency, into a sort of very rapid, very convenient library construction protocol. And I'm going to end this part of the series just with a few data slides, and I wanted to show you this just to show you what a big difference library construction can make to the outcome of your to the library quality and the outcome of your sequencing in the end. And so what we're looking at here are called GC bias plots, really, or coverage uniformity plots. What we often do, us and others, is to use microbes with different genomic GC content as sort of models for looking at bias and coverage uniformity. So on the one end of the spectrum, we have Clostridium that has a 29% GC content, so it's 80 rich bacterium. E. coli has pretty much got a balanced GC content, or average, or half, just over half G's and C's and half A's and T's. And then Bordetella pertussis is a pretty GC-rich organism, so almost 70% of the genomes are G's and C's. And so what these plots do, if you look at the gray bars in the back, is it essentially takes the whole genome and it sort of bins it, breaks it up into 100 base pairs, just from start to end, and then it puts these 100 base pairs, basically arranges them in order of GC content. And so, Clostridium, you'll see that the average, the average is around here, 29, 30%. And so the height of the bar represents how many 100 base pair windows falls, in this range of, 11%, 12%, 13%, 14, 15% in each case. And so, as you can see, as the GC content increases, the sort of mode of this peak shifts to the right and to the right. So if you look at the axis at the bottom, the GC percentage is not always the same. It's going to go from 10 to 50 for the 80-rich organism, 25 to 70 for the more balanced organism, and then 45 to 85 for the GC-rich organism. And then what we've done is made libraries with different types of library construction strategies, so representing the things that we just discussed. And so the two blue lines are using the Kapa HyperPrep kit. So this is our streamlined library preparation, ligation-based library preparation kit, either in combination with mechanical shearing, which is Kapa shearing, or enzymatic fragmentation, the new reagent that we've just launched. And so those are the blue lines. The orange line, and sorry, I apologize that the color distinction isn't that great. So the darker orange line is a competitor kit that also uses ligation-based library preparation with a different type of enzymatic fragmentation. And then the very light line is a fragmentation-based library preparation kit. And so what we're looking at is normalized coverage, essentially, and coverage uniformity. So if everything was perfect, if library construction was perfect, cluster amplification was perfect, and sequencing was perfect, we would expect every single one of these sort of GC bins to be represented with the same coverage depth, so the same number of reads across. And so this normalized coverage, essentially says, so what is the coverage of this bin relative to the mean coverage for this entire genome? And so, as I said, in a perfect world, we would expect, a straight line at normalized coverage of 1. And so when we look at the AT-rich organism, and we look at the coverage uniformity obtained, or the coverage, normalized coverage obtained with the Kapa kit, whether it was enzymatic fragmentation or enzymatic fragmentation, it's pretty darn close to that, to a perfect sequence. When we look at the other method, you can see that there's huge biases. So, in this case, there's a huge underrepresentation of AT-rich sequences, very highly AT-rich sequences. There's almost no Gs at this point, and an overrepresentation. So in AT-rich organisms, the very AT-rich stuff, the extreme AT-rich stuff is the difficult stuff. And so the more GC-rich, which isn't really GC-rich, it's only like 30 or 40% GC, is overrepresented because it's easier in a way. And so what this means in the end is, to obtain the same sort of coverage depth, you have to do a lot more sequencing to get these underrepresented sequences, right up there. And so, for the AT-rich organisms, not so bad when we kind of go through, even E. coli has really difficult sequence in it. You can see the blue line, so the Kappa hyperplus and Kappa hyperpre plus Kavaris line is a little bit more skewed to a little bit of overrepresentation of things in the easy range, which is 30 to 40% GC. Little bit less representation in the GC, so not perfect, but still a lot better if you look at the other library construction methodologies, how much more these regions are underrepresented. So again, it means a lot more sequencing to get these regions, up there to where they're all uniformly covered, or at least meet the minimum coverage specifications. And then the same sort of story with the GC-rich organism. The lines just tilted the other way, because the relatively easy stuff is, is now in the lower end of the GC spectrum, and the difficult stuff. And when we get here to sort of 75 to 80% GC-rich, that's just where the sequencing chemistry fails entirely, and it's really, really difficult to obtain any sequence data. But so I wanted to leave you with this sort of very, clear picture of how library construction, how the methodology that you choose can affect the outcome of your library and the quality of the library, and that impacts in the amount of sequencing you have to do. It impacts sequencing cost, and it also impacts sequencing time. So, whether you're a PhD student and have a project to complete, or whether you're a clinician, or doing clinical sequencing and you have patient results to get out, it matters. And so making the best possible library is really what we want to do to ensure that we get basically the best bang for our buck when we sequence. And so in summary, we asked the question in the beginning, what are the objectives of library construction? In a practical or physical sense, library construction means we have to fragment DNA and get adapters on them in some way or another, so that we can do cluster amplification and sequencing and multiplexing. The more philosophical objectives is we have to make the best possible library we can. And so that means, that library that has the most unique fragments in it, so that you have the highest coverage depth. But it also means that library that has the most uniform coverage, so it has the similar number of unique fragments covering every single position, so that you can do the least sequencing to achieve your goals. And so to achieve that, you need to select a method or a protocol that ensures the lowest fragmentation and amplification bias. And as far as fragmentation technologies go, Kavaris shearing or the new Kappa frag enzymatic fragmentation kits are the best performing ones. You want to use the library preparation protocol that gives you the best library yields. We believe ligation-based protocols are the best ones, and we'll talk about that in detail in the next part. And then, also obviously, performance is really important. But in the end, if everything's all equal, you want to pick something that's fast and convenient. And so that's really what we've tried to achieve with the Kappa Hyperplus kit. It's a very high-performing library preparation kit that's really, quick and easy. And allows you to, to achieve the sort of throughputs that you need, whether you're, a single person that needs to make 100 libraries, or a big lab that makes hundreds of thousands of libraries. A quick library preparation kit that performs really well is basically the best solution.